Sun's Lab. It's my pleasure today to introduce our speaker, Dr. Sheikh-Hamad. According to the CDC, 1 in 10 Americans, about 20 million, are living with some kind of chronic kidney disease. The hospitalizations due to acute kidney injury have risen from 4,000 in 1996 to over 23,000 in 2008. Today's speaker, Dr. Sheikh-Hamad, is a physician scientist who is in kidney disease. Dr. Sheikh-Hamad received his bachelor's degree from the University in Jerusalem and completed his medical training at Hadassah Medical School. He subsequently pursued advanced training in nephrology at UCLA. Currently, Dr. Sheikh-Hamad, the professor in medicine at Baylor College of Medicine, section chief of renal service at Ben Taub Hospital, which is Houston's biggest public hospital. Besides his demanding clinical responsibilities, he also directs active NIH and VA-funded research in his laboratory. He has published over 70 papers, and his team has made several discoveries on multifaceted roles of semiocalcin, one in kidney functions. Dr. Sheikh-Hamad has been given numerous honors and awards. Besides many prestigious academic awards, as a student, I am very happy to know that he is also the recipient of the Faculty Excellence Award and Distinguished Educator Award. Dr. Sheikh-Hamad's lecture today is Mitochondrial Pathway for Kidney Protection. So please join me to welcome and thank Dr. Sheikh-Hamad.
Thank you for the kind introduction. So let me tell you something to really impact upon you, a story that not many of us are familiar with. And I give this story to the medical students every time. And the question is, how many Americans are there with chronic kidney disease stage 3, 2, and 3? Normally, we define kidney disease based on stages. Normal is 0, stage 0. 1 is some kidney disease, but the GFR or the filtration rate is not affected that badly. 2 and 3, essentially about 50% of the kidney function is gone. And 4, you're down to about 25 or 30% of the kidney function. 5, that's when we put patients on dialysis. So if I told you that there are about 20 million or 25 million Americans with chronic kidney disease stages 2 and 3, and about 500,000 Americans with stage 4 kidney disease, and about 500,000 Americans on dialysis. Question number 1, wouldn't you expect those patients with chronic kidney disease stage 2 and 3 to progress to 4 and 5, right? Wouldn't you expect these patients to be on dialysis? That is 20 million Americans on dialysis, right? You would. You would. The answer is, you don't see them on dialysis because they die. They don't make it to actually start dialysis. So the message from this little story is that chronic kidney disease is a major risk for cardiovascular disease. Most of these patients actually die from cardiovascular disease. So with that opening, which is a bit depressive, I will tell you about our work and why we do what we do. I've been working on this topic now for nearly 7 or 8 years. And this particular molecule, as you will see soon, led us to some interesting discoveries which potentially hold the fountain of youth. And I will let you judge for yourselves. So look at these numbers. About 5 to 10% of hospitalized patients will develop renal failure. About 70% of the patients in the ICUs will have renal failure. And about 5-6% of these patients will require dialysis while in the ICUs. So, and once acute kidney injury has occurred, the treatment pretty much is supportive. In spite of millions of dollars put into research to find out cures and treatment for acute kidney injury, we are not wiser today than we were 20 years ago. So here are some numbers to impact upon you how bad having kidney disease is. So normal creatinine for the average person in the crowd here is 0.8 to 1 milligram percent. So that's 1 milligram per 100 milliliters of blood. A rise in serum creatinine of about 0.5 milligram percent will lead to an increase in odds of death of about 6.5-fold, an increase in cost of care of about 7,500. Look at these numbers. These are adjusted in the blue bar. This is adjusted for covariables, and an increase in creatinine, serum creatinine greater than 2, will be associated with an odds of death increase of about 50-fold, an increase in the cost of care by more than $20,000 to $25,000, and of course, length of stay in the hospital will increase dramatically, and that too costs money. So the impact is huge. Not only that, so if we look at acute kidney injury event, so many patients here in the red line will go directly into dialysis. They will not recover from kidney injury. Some patients who had chronic kidney disease before, if you look at this solid dark green, will develop acute kidney injury and then recover a little bit, but go into an advanced stage of moving towards dialysis. Others will start with some degree of chronic kidney disease here in the light green and have some acute kidney injury, recover, and go on the same slope of developing or progression towards end-stage renal disease. And some will recover completely after the insult. So how do you differentiate between these? And that's what we're trying to learn. So our research has, over the last 10 years or so, focused on acute kidney injury. And acute kidney injury model in the lab is induced by clamping of both pedicles, which means the renal arteries and veins together, bilateral, clamping for about half an hour. This induces ischemia for half an hour. And once you open those pedicles, you restore blood flow. That's when the real injury occurs. That's when you activate reactive oxygen species and inflammation. So experimental ischemia-reperfusion kidney injury is a close mimic of ischemic AKI, acute kidney injury in humans, and has been utilized to study the pathogenesis of ischemic AKI. ATP depletion, reactive oxygen species, inflammation, and endothelial barrier dysfunction play important roles in the pathogenesis of ischemia-reperfusion kidney injury. Stanicalcin, a protein, actually a hormone, which was discovered initially in fish, named after the discoverer, Stanius, German scientist, and calcin, because it regulates calcium in fish, hence staniocalcin. And in the fish, feeding and swallowing aquatic environment and containing the food, or the aquatic environment exposure directly to the gill. These are the two organs that are continuously exposed to the aquatic environment. In case that fish swims in high calcium content, then calcium floods the bloodstream and can induce cardiac arrest. So the fish has designed, or nature has designed, a system whereby calcium entry through the aquatic environment, through the gill and the intestine, would be regulated. So upon rise of calcium concentration in the blood of the fish, this hormone is released from a small tiny gland, accessory organ to the kidney in fish, called the gland of Stanius. And that hormone, upon circulation in the intestine and the gill, it blocks calcium channels, blocks calcium entry into the circulation. The mammalian homologue of this fish hormone has evolved evolutionarily and does different things, although at the cellular level it still regulates calcium channels, and we have shown that in cardiomyocytes, where it actually works pretty much like a blood pressure medicine, the calcium channel blockers, L-type calcium channel blocker. I will emphasize this point. It's an intracrine protein. An intracrine protein is a hormone that binds to a cell receptor at the cell surface, activates signaling from the cell surface, and then ends up going intracellularly, either ending up in the nucleus or in the mitochondria or in other organelles, and it acts there in addition to its action from the cell surface by inducing signaling there. So here is an example of what happens to staining staining. So this is macrophage, and this is confocal image, and DAPI staining for the chromatin here, which is really not uniformly dense. Red here is staining. We add staining to the medium, recombinant protein, and then wash the cells and then subject them to staining. And as you can see, plenty of red within the cell. So entry of staining into the cell. And where does it go? It goes to the mitochondria. Here green, mitotracker, and red and green combined produce yellow. So association and co-localization of staining in the mitochondria. So protein that we added to the cell surface ended up actually in the mitochondria. So upon activation of signaling from the cell surface, which I will show you later, this protein ends up within the mitochondria. So what does this protein do? And on the left side here, mostly work from our lab. Decreases superoxide generation in macrophages, endothelium, cardiomyocytes. The effect is predominantly UCP dependent, uncoupling proteins that is dependent. And potentially SIRTUIN3. SIRTUINs are longevity genes, and I will present some additional data about this later on. Preserves the expression of endothelial tight junction proteins and stabilizes endothelial barrier function. Inhibits transendothelial migration of T cells and macrophages, and in a model of anti-GBM, anti-glomerular basement membrane disease, which has the human equivalent, it actually protects from this disease. So antibodies that attack the GBM, the glomerular basal membrane in the kidney. Other effects, although not from our lab, reported by others, enhances phosphate uptake. This is whole animal study. Effects on bone and skeleton, angiogenesis, reproduction. There's high level of staining in the circulation of pregnant mothers, as well as in the milk. So during lactation, the amount of staining in the circulation and in the milk increases significantly. And there's some science about that as well. And carcinogenesis, but we will not be touching upon this today. Here is mitochondria 101, and in fact, when I made the initial discoveries about the involvement of staining in the mitochondria and went to Rice and sat with the students there to relearn the mitochondria. And here's what I learned. So if you look at the mitochondria on here, and this tiny box is enlarged, and this is the outer membrane, and this is the inner membrane, and what you see there in yellow, this is the matrix, and that's where the Krebs cycle enzymes and the fatty acid oxidation occur. High energy molecules are generated from the metabolism of the fatty acids and the Krebs cycle in the form of NADH and FADH2. These are being utilized by the respiratory chain complexes to essentially generate ATP. What happens? Electrons are moved from one complex to the other with the potential of meeting atomic oxygen and generating reactive oxygen species, here superoxide. At the same time, hydrogen is being pumped from the matrix into the intermembrane space to generate high concentration of hydrogen here and an electrochemical gradient favoring hydrogen movement from the intermembrane space back to the matrix. This is normally happening through the ATP synthase, and in the process, the ATP synthase generates ATP from ADP. Now think of hydrogen here, concentration being higher in the intermembrane space as compared to the matrix. Think of it as water above the dam, and when it goes down, its electrochemical gradient, it drives ATP generation. There's a problem with this, however. The more hydrogen ion transport there is to the intermembrane space, the higher the membrane potential. And the higher the membrane potential, the more superoxide you generate. and that's not good. So it can be injurious to the cell and essentially lead to apoptosis. Nature has devised a safety valve for this. And the safety valve is this uncoupling protein channel, which allows hydrogen to come back to the matrix, bypassing the ATP synthase. But in the process of bypassing the ATP synthase and allowing hydrogen to come back, it decreases membrane potential and decreases reactive oxygen species. So the gain here is we reduced reactive oxygen species at the price of slight reduction in the generation of ATP efficiency. So decreased ATP efficiency, ATP generation efficiency, and the gain is reduction in superoxide generation. And here's what happens to macrophages treated with stenocalcin. Uncoupling protein 2 abundance increases within 2 to 3 hours, about threefold. And as you would expect, decreases membrane potential here if you look at the white bars. And as expected also, decreases superoxide generation. And if we take macrophages from UCP2 knockout animal, where the macrophages are knocked out for uncoupling protein 2, the effect of stenocalcin is diminished or abolished. So the effects of stenocalcin to reduce, the effects of stenocalcin to reduce reactive oxygen species or superoxide are mediated in great part through the upregulation of uncoupling protein 2. So is this relevant biologically? Yes, it is. Here are macrophages, wild type, treated with stenocalcin, and the white bars here, the reduction, nearly half reduction of superoxide generation at baseline, not stimulated. If you stimulate with LPS, again, increase in superoxide generation, that too is suppressed by stenocalcin. So suppression of LPS-induced reactive oxygen species. And this is very important because, as you know, LPS is a modulator of the innate immune system. So here you have, in the form of stenocalcin, a modulator of the innate immune response. Now, if you take, we learned in my biology and medicine classes that the bulk of reactive oxygen species in macrophages are generated by the NADPH oxidase system. So we took macrophages that are deficient in one of the subunits of NADPH oxidase, and that is GP91 FOX. So it doesn't have NADPH oxidase activity for the most part. And here too, we see quite a bit of reactive oxygen species being generated in spite of the absence of NADPH oxidase, and that too is suppressible by stenocalcin. There's also upregulation of reactive oxygen species by LPS, and that too is suppressible by stenocalcin. And what if I told you that LPS increases the generation of reactive oxygen species in macrophages by actually decreasing the abundance of uncoupling proteins? So uncoupling proteins participate in the response of LPS and modulation of the immune system, and stenocalcin is a natural antagonist for LPS-induced superoxide generation. So here's some additional evidence. Mitochondria from UCP2 knockout macrophages produce more superoxide and hydrogen peroxide, and macrophages from UCP2 knockout mice show increased NF-kappa B activity. This is a major pathway to activate inflammatory response in macrophages, and they also phagocytose pathogens as if there is no tomorrow, enhanced phagocytosis. So here, reactive oxygen species generation through a pathway in the mitochondria regulated by uncoupling proteins is important for biology. What else? So here is a simple assay. We take endothelial cells cultured over an agarose bed. And then when you treat endothelial cells with a cytokine, you open the tight junctions between neighboring endothelial cells, and they become more porous and permeable. So when you treat here with TNF-alpha, the permeability for FITC dextran, high molecular weight dextran labeled with FITC, so increases, doubles. If you treat with stannicalcin by itself, nothing happens. But if you treat with both at increasing concentrations of stannicalcin, you block the effects of TNF-alpha on the permeabilization or increasing permeability of this monolayer of endothelial cells. The same thing happens if you place macrophages or T cells on top of this monolayer of endothelial cells. So treated here with interleukin-1 beta to open those tight junctions between neighboring cells, normally would increase the migration of T cells and macrophages here in the black bars. And as you increase the concentrations of stannicalcin in a dose-dependent manner, decreases the migration of T cells and macrophages. So here I've showed you a protein that inhibits macrophages, stabilizes endothelial barrier function, right? And I've told you before that these are key in the pathogenesis of acute kidney injury, ischemic acute kidney injury, and it's reasonable to hypothesize that stannicalcin would have some anti-inflammatory and protective properties in the setting of ischemia reperfusion kidney injury. Make sense? And I just told you this. So we took mice that were generated by a collaborative group in Canada. Here is human stannicalcin driven by the metallothionein minimal promoter. And you predict that it would have global overexpression of stannicalcin. However, these mice show preferential expression of stannicalcin in endothelial cells, blood vessels, and certain organs. And as you can see here, these mice are smaller in size compared to the wild-type littermates. The transgenic mice are 30 to 40% smaller in size, consume more food and oxygen per gram weight, clear glucose faster, and have thin fat pads, the magic bullet for the overeating American, the average American. So high serum levels of stannicalcin and normal serum calcium, creatinine, blood pressure, and hematocrit. So wonderful medicine to take. And if you look at the kidney, and if you look at uncoupling protein staining, this is wild type and these tubules, and this is uncoupling protein fluorescent staining. There's increased abundance of this uncoupling protein 2 in the stannicalcin transgenic kidneys. And this is essentially live staining for superoxide using mitosox, substance that goes to the mitochondria and interacts with superoxide. And when it does, it fluoresces red. And then fix those sections and mount them on the microscope, and you have these beautiful images. And as you can see, kidneys of stannicalcin transgenic mice generate pretty much less superoxide, as we have shown you in macrophages. So let's apply now this model of ischemic acute kidney injury to mice. And to remind you, we are clamping both pedicles, that is renal arteries and veins, on both sides for 30 minutes and then release. And then 24 hours later, we sample blood for creatinine clearance measurement, which is the function of the kidney, and take out the kidneys and stain them to look at the morphology. And as you can see here in the black bars, kidney function is about 30%, 50% lower by this 48 hours time point. Sustained reduction at 72 hours, and as predicted, recovery of kidney function by 8 days or 10 days in this model of ischemic acute kidney injury. And as you can see in the white bars, these are stannicalcin transgenic mice subjected to the exact same insult. Nothing happens to kidney function. Now, these actually tell the story better. Look at these pinkish structures. This is the brush border in the proximal nephron. And these are the villi to increase the surface area for absorption in the proximal nephron. And they stain nicely and positively with PAS. These are the first to disappear after ischemia reperfusion kidney injury. Subjected to ischemia, gone. And as you can see here, they're gone 24 hours after the insult in wild-type mice. And dilatation of the tubules, cast formation. These are cells that have died, sloughed, and now blocking the pathway of urine in the tubules. Inflammation here, as you can see, spotted in the interstitial compartment at 48 and 72 hours, and recovery, and look at the brush border membrane in this model, recovery of kidney function 8 days later. Look down here. These are Stennicalcin transgenic mice subjected to the exact same injury, at least subjected to the exact same treatment vis-a-vis blocking of arterial and venous pedicles. And as you can see here, the brush border membrane is intact. Absolutely nothing happens. And we'll whiz through these quickly. So what happens in the wild type? Increased superoxide generation after ischemia or perfusion, increased hydrogen peroxide generation, and increased permeability as measured by Evans blue penetration of the kidney tissue. And then we harvest the tissue and measure Evans blue presence within the tissue. So a measure of vascular permeability. And here, look at CD3. These are T cells, increased abundance in black bars here in the wild type, as well as macrophage increase in the tissue. So whereas the Stennicalcin transgenic mice show none of these changes, no increase in superoxide generation, hydrogen peroxide generation, no increase in permeability, vascular permeability that is, and no increase in T cells or macrophage infiltration in the kidney. So we'll just move on to the next portion of this presentation, and that is the link with uncoupling proteins and SIRTUIN-3. In adipocytes, UCP1, as you know, UCP1 is responsible for the shivering phenomena and the generation of thermogenesis, right? Heat from food that we eat, and that is through increased abundance of uncoupling protein 1 in the brown adipose tissue. That's how the mouse in the desert at night heats itself. Stennicalcin 1, SIRT3 and UCPs localized to the inner mitochondrial membrane. This is something that I didn't show you, but these are facts. So inner mitochondrial membrane. So we hypothesized that there might be a link between these three molecules, functional link that is. A word or two about SIRTUINS. Null mutation in SIR2, silent mating type information regulator. That's the name that was given to the first representative of this family of proteins by a yeast biologist, mucking around with yeast genes, deleting and duplicating, and that's what was discovered. So mutation in this gene shortens while extra copy of this gene prolongs lifespan by about 50%. SIRTUINS localized to the nucleus 1, 6, and 7, mitochondria 3, 4, and 5, and cytoplasm SIRTUIN2. Akin to phosphorylation, dephosphorylation. So you have the kinases phosphorylating proteins or enzymes to activate in general, but that's the rule. Or dephosphorylating to inhibit in general, but that's not the rule. So akin to phosphorylation, dephosphorylation by the kinases, phosphatases, respectively, SIRTUINS acetyl transferase or acetyl transfer, a moiety of acetyl that is tagged to lysines on proteins. So tagging or untagging essentially activates or inhibits proteins within the mitochondria, pretty much similar to what the kinases and the phosphatases do. Generally, untagging the lysines or taking off the acetyl moiety from the lysines is activating, whereas tagging with lysine is inhibiting. But again, that's not the rule always. Look at this. Nearly 30% of the 1,200 known mitochondrial proteins are regulated by SIRTUINs, mainly SIRTUIN3, because if you knock out SIRTUIN4 and SIRTUIN5, pretty much nothing happens to the acetylation profile of mitochondrial protein. Activation of SIRTUINs increases resistance to metabolic, oxidative and hypoxic stress in different tissues. And in general here, so proteins where the acetyl moieties were chipped off and activation presumably, and in an NAD-dependent manner, which means these molecules are energy sensor and they do this acetylation, deacetylation depends on their perception of the energy state of the cell. Activation of sirtuins will inhibit senescence, promote survival, DNA repair, metabolism, proliferation, and of course the NAD to NADH ratio, higher NAD plus to NADH ratio, will promote activation of the sirtuins. So you want to have a lot of these, guys, especially when you age, because specifically sirtuin 3 has been linked to longevity in humans. So is there a linkage between stennicalcin and sirtuins? And here we did an expensive experiment, really. We made synthetic peptides derived or based on the sequence of stennicalcin. 17 amino acids each peptide, hopping along the sequence 2 amino acids each time. So we covered the entire sequence of stennicalcin, and then these peptides were dotted on a PDF membrane, as you would be doing for Western blot, and then incubate with sirtuin 3, and then follow up with anti-sirtuin 3 antibody and reaction. As you can see here, there are certain regions within stennicalcin that interact directly with sirtuin 3. So there's direct interaction between stennicalcin 1 and sirtuin 3. Not only that, when we express stennicalcin and sirtuin 3 and then pull with here flag stennicalcin, pull down flag, we discover essentially sirtuin 3 within the immunoprecipitate. And not only that, if we co-express stennicalcin with UCP1, UCP2, UCP3, we detect all of them in the immunoprecipitate. So apparently there is a complex of stennicalcin, sirtuin 3, and all of these uncoupling proteins that I mentioned to you. There are also other uncoupling proteins in the brain, 4 and 5, and probably unknown number of other uncoupling proteins in other tissues. But the main uncoupling proteins, 1, 2, and 3, obviously associate with sirtuin 3 and with stennicalcin, form a complex presumably within the mitochondria at the inner mitochondrial membrane, and somehow regulate reactive oxygen species. Not only that, stennicalcin 1 knockout mice have less abundance of sirtuin 3, so regulation of sirtuin 3 directly by stennicalcin. And in addition, the profile of acetylated mitochondrial proteins is also different, which means stennicalcin regulates the abundance of sirtuin 3 as well as its activity, presumably. And not surprisingly, if you look at the abundance of uncoupling protein 2 in the kidney tubules, you will find that there's upregulation of uncoupling protein 2 in the tubules of stennicalcin transgenic kidneys, as you would see in the tubules of stennicalcin 1 transgenic kidneys, and again, decreased reactive oxygen species. So whether you upregulate this or upregulate this, both will lead to the same result, and that is reduction of reactive oxygen species generation. Now, let's move to the last portion of this, actually one before last, there's another one, and that is the AMPK link between these proteins. And there have been reports that activation of AMPK, be it using ICAR or metformin. Metformin is a medicine used to treat diabetes type 2. ICAR is a medicine that is used in certain cultures. I was told in China it's being used as a modulator of diabetes therapy, but I'm not sure. So we hypothesize that stennicalcin suppresses oxidant stress and protects from ischemia, reperfusion, kidney injury through activation of AMPK. Is that true? And let's talk a little bit about AMPK. So again, another energy sensor, an important molecule in every cell in our body. It's ubiquitous and highly expressed in the kidney. It's a metabolic sensor that regulates energy generating and energy consuming pathways. During energy stress, when ATP decreases and AMP concentration increase, then AMPK is activated, and what it does, it phosphorylates multiple substrates that stimulate energy production, fatty acid oxidation, for instance, glycolysis, and minimizes energy consumption. So essentially recalibrating energy utilization in the cell. It has an important role in the normal physiology and pathology in the kidney. So if you look at these are major transporters and energy consuming transporters, either directly or indirectly in the kidney, such as the CFTR, the ENaC, which is the major sodium channel in the principal cells of the collecting duct, the NKCC, which is the sodium potassium 2 chloride, key transporter for sodium and chloride in the thick ascending limb, which is the target of Lasix, by the way, the diuretic Lasix. And hydrogen ATPase in the intercalated cells, which is responsible for hydrogen homeostasis. Reduced AMPK activity in the diabetic kidney is associated with accumulation of triglyceride and glycogen and leads to kidney hypertrophy. So activation of AMPK normally happens when the AMP to ATP ratio increases. And again, it's an energy sensor. So activation of AMPK is triggered by changes in energy homeostasis. And when we look at standing calcium transgenic kidneys, and lo and behold, indeed, AMPK, this is phospho-AMPK. And phosphorylated AMPK is at higher abundance in standing calcium transgenic kidneys. And these are the quantitation. We had some compulsive reviewers reviewing this paper. They wanted phospho-AMPK alpha 1, alpha 2 over total AMPK alpha 1 and alpha 2. They wanted that 2 over actin. The bottom line is there's upregulation of AMPK or hyperactivation of AMPK in the kidney of standing calcium transgenic mice. And look at this one here. This is alpha 2. It appears that the alpha 2 is predominantly or considerably more activated than the alpha 1 in these transgenic kidneys. But this really tells the story here. We took a number of tissue sections here on the left side from standing calcium transgenic kidneys, wild type, standing calcium 1 knockout, and standing calcium 1 and 2 knockout. And as you can see, AMPK activity nearly disappears once you hit the double knockout of standing calcium. Conclusion is standing calcins in the kidney are major activators of AMPK, and I would not be surprised to find out that standing calcium is a key activator of AMPK in other tissues, which makes it really very important. And this is quantitation here at the bottom. And again, to convince the reviewers that we are not imagining stuff, we took hex cells. Hex cells were taken from an embryo that was aborted by, I think, a Dutch mother at an early stage in the pregnancy. And these cells were transformed somehow and cultured in vitro, and they have been maintained through generations. So kidney cells, the origin is debated, but nonetheless, a pretty good model to study responses in the kidney. And as you can see, treatment of these cells with standing calcium within 15 minutes activates AMPK. Look here. So, and this is the dark black graph here. And no change in the total AMPK, so no change, and no change over actin. And if you look at SIRTUIN3, there's some delay in the upregulation of SIRTUIN3 protein. So abundance of the protein increases. Similarly, there is increase in the abundance of UCP2. So in vitro, when we treat these cells with standing calcium, we activate the AMPK, and following the activation of AMPK, there's an increase in the abundance of SIRTUIN3 as well as UCP2. So that wasn't convincing enough, so we went ahead and treated these cells. First, we treated with stanicalcin in the presence of an inhibitor of AMPK, which is a CC compound. And as you can see here, inhibition of phosphorylation of AMPK, and when we do so, in spite of the treatment with stanicalcin, inhibition of AMPK blunts the upregulation of SIRTUIN3 as well as UCP2, indicating that upregulation of UCP2 and SIRTUIN3 by stanicalcin are dependent on AMPK activation. So without AMPK activation, that does not happen. And these bars here, these graphs, are essentially quantitation of what I just told you. What happens in the stanicalcin transgenic mice? This is not the best representative blot, but as you see here, when we inhibit AMPK with CC compound, and this is quantitation, you diminish the expression of UCP2 as well as SIRTUIN3. So in vivo, in the stanicalcin transgenic mice, when we inhibit AMPK, we decrease the abundance of these two proteins, consistent with our in vitro data that active upregulation of UCP2 and SIRTUIN3 by stanicalcin are mediated through activation of AMPK. So what happens now to the response of the kidney to ischemic injury when we inhibit AMPK in stanicalcin transgenic mice? And look at this. So as I showed you before, these mice did not budge to an ischemic injury of 30 minutes clamping of bilateral pedicles. And so here, vehicle, CC compound, or the inhibitor of AMPK, be it, so this is in the sham, and this is with ischemia reperfusion. And as you can see, when you inhibit AMPK, you now recapitulate the responses that we saw in the wild type mice, and that is increase in reactive oxygen species generation. But not only that, we also recapitulate the injury. All of a sudden, these mice show susceptibility to ischemia reperfusion injury. They increase the serum creatinine, they decrease the filtration rate and urine output. So they develop renal failure when you inhibit AMPK. So again, the protection through overexpression of stanicalcin is mediated through activation of AMPK and presumably through upregulation of protective molecules such as UCP2 and SIRTUIN3. So we have this scheme to explain how stanicalcin works. So it binds to the cell surface, activates AMPK, so signaling from the cell surface, and true to its nature as an intracrine molecule, it ends up in the mitochondria. And what it does in the mitochondria, we know that it associates with SIRTUIN3 and UCP2, and it leads to decrease in mitochondrial membrane potential, decrease in reactive oxygen species, but also we believe that there are some changes in metabolic pathways that are regulated through changes in SIRTUIN3, and that's what we are looking at right now. Now finally, before we finish, and I wouldn't take much time, I'll finish in time for some questions. What happens if you knock out stanicalcin acutely in the normal kidney? So we devised a nifty experiment to answer this question. So the design was based upon generation of transgenic mice again, and the transgenic mice essentially express a cassette for shRNA, be it stanicalcin-1 shRNA or scrambled sequence shRNA, and separated this cassette from the H1 promoter by a stuffer which has a reporter gene, PGK promoter-driven EGFP. So these mice fluoresce green, so we can detect them easily. And if you do Cre recombinase treatment, these are essentially removed, and you bring the H1 promoter upstream of the cassette to either produce stanicalcin-1 shRNA or scrambled sequence shRNA. And I will skip these. These are essentially the controls for the reviewers to be satisfied. And again, recombinase removes this, brings the promoter, H1 promoter ahead of the shRNA cassette, and the shRNA produces either stenocalcin-1 shRNA or scrambled shRNA. And then we have phenotype. And these are the basic controls for the experiment. This is stenocalcin staining, so it's present everywhere in the kidney. Literally every cell in the kidney expresses it. And this is staining control, so negative control. And this is scrambled shRNA transgenics, so treated with the Cre. And how did we deliver the Cre to knock out stenocalcin specifically in the kidney? So we had Cre recombinase driven by a promoter. We tried Tie2 Cre, which is specific to endothelial cells. We tried also CMV, we tried also KSP promoter, which is specific for the tubule cells. And the idea here is you have a plasmid that we wrap it in lipid liposomes, essentially, mixture of phospholipids, albumin, lipofectamine, and some other goodies with perfluoropropane gas, which the ophthalmologists use to buckle the retina when they do surgery that requires delicate handling. And so the bottom line is now we have vesicles that contain this plasmid. And we inject the plasmid through the tail vein, and it circulates, and we blast the kidney with high energy ultrasound to essentially disperse these microbubbles and transfecting the kidney cells non-selectively. All cells within the kidney are transfected with the plasmid that expresses the Cre, and essentially to activate the shRNA cassette, the stenocalcin-1 shRNA or the scrambled shRNA. So here's in situ hybridization of scrambled shRNA transgenic mice treated with the microbubbles and with the delivery of the plasmid. And as you can see, a lot of brown here, essentially positive staining with the anti-sense probe for stenocalcin. And here with the sense probe, so to show you that the staining was actually pretty good. And when we knock out or knock down the stenocalcin here with the anti-sense probe in stenocalcin-1 shRNA transgenic mice, we essentially pretty much eliminate the expression of stenocalcin-1 shRNA. And this is global view of the kidney using an antibody that highlights stenocalcin expression in blood vessels. And why do you see these rays? These are the medullary rays within the medulla. These are blood vessels that traverse the kidney medulla down. And what are these dots? These are glomeruli rich in capillaries. So you see them nicely, and they are pretty much gone in the shRNA transgenic, stenocalcin-1 shRNA transgenic. And here another staining just to show you it's gone. 4 days after treatment, and this is 5 days after treatment compared to the controls. And trust me, this is quantitation of the messenger RNA, the protein, and the staining, and they're all down. And what happens, as expected by now, we would expect to see less uncoupling protein expression. And indeed, there is less uncoupling protein expression and more reactive oxygen species. But not only that, surprisingly, now we see some interesting phenotype, and that is all of a sudden vacuolization in these cells, in the shRNA, stenocalcin-1 shRNA transgenic kidneys, when we knock down stenocalcin in an acute fashion. Vacuolization. And the vacuolization takes place here in cells that express aquaporin-1. Where is aquaporin-1 expressed in the cortex in the kidney? In proximal tubules. So by definition, the aquaporin-1 site of expression is the proximal nephron in the cortex. So you see the vacuolization in aquaporin-1 expressing cells, but not in aquaporin-2 expressing cells, which happen to be in the distal nephron collecting ducts. And associated with apoptosis here, staining. But not only that, so we see activation of XBP1, which is the unfolded protein response. So the cells are really stressed badly. And conversion of LC3-1 to LC3-2, indication that there's also activation of autophagy, which in our opinion here is a pathway to death. And if you look at the mitochondria here, the mitochondria look sorry. They're inclusion bodies, they're condensed, they're small, as compared to the nice hustra and size of these mitochondria, similar magnification. So there's big time injury, which then ends up with this final phenotype, and that is decreased creatinine clearance, increased serum creatinine. So knockdown, acute knockdown of stannicalcin in the kidney without any additional injury by itself will lead to acute kidney injury. And the conclusion is stannicalcin is a significant component of the kidney's antioxidant defenses under normal physiologic conditions. A knockdown or knockout of stannicalcin will by itself lead to acute kidney injury without even needing another insult. And thank you for attention. And without these and their contribution, this work would not have been done. And also many thanks to the NIH and VA for their support. Thank you.
That's time for questions. Try to make questions shorter. We have 5 minutes.
Yes, ma'am.
Jenny, please.
So has the receptor for STC1 been identified?
There's indirect evidence for its existence, and this Canadian group with which we collaborate has generated a chimeric stannicalcin alkaline phosphatase protein. And what they do, they assay alkaline phosphatase activity. And they have shown using membrane preparation that when you use this chimeric protein and then follow up on it with competing non-alkaline phosphatase chimera stannicalcin, there's completable, saturable, inhibitable, saturable KM binding of this stannicalcin alkaline phosphatase to mitochondrial as well as plasma membrane. No protein in hand, no gene identified yet.
Well, question?
So I was just wondering, you know, in a very simplistic way of thinking, things like caloric restriction, which would increase AMPK, is there an epidemiology or something? There's less kidney disease in people. Well, I would suspect that obesity must be a contributing factor to kidney diseases. Is that true?
Well, obesity is a contributing factor to kidney disease, and there's a specific kidney disease, which is focal segmental glomerulosclerosis, that happens in obese subjects, especially if there's a component of hyperlipidemia at the same time. Stannicalcin, we're exploring the question that you asked. It turns out it has significant contribution to the survival of transplant organs and some other protective properties in models of injury to other organs, including lung, for instance, from bleomycin. Upon injury to the lungs with bleomycin, the injured lung calls in mesenchymal bone marrow stem cells that provide protection to the injured lung through upregulation of stannicalcin. So it's a naturally employed defense system, not only in the kidney, it's employed in other organs in response to other injurious insults. But it also has, now we're finding out as we are exploring protection by stannicalcin in other models of disease.
Any other questions?
Just follow up Champion's question. So in type 2 diabetes, like a renal failure is common. So how hyperglycemia or insulin resistance can promote kidney failure?
We're looking at that right now. So I'm fortunate enough to have underneath the floor where my lab and office is, an endocrine department with very bright people. So I'm now addressing exactly this question with one of them. Yes, sir.
In your EM picture, when you knock down your protein, do you see the morphology of epithelial cells being affected?
When we knock down, yes.
Yeah.
Yes, and in fact, there's a follow-up to this story, which is not fully cooked yet. If you invite me again within a year, I will have the full story wrapped up. And I'll be happy to tell you a little bit more about this pathway than I have, specifically about this. This how has now been figured out. We're just ironing out the last pieces, and this turns out to be a very interesting box of the puzzle. So invite me again in a year and I will tell you more about it.
So due to timing, so I just like to stop here and thank you for just thank our speaker for a great talk.
Thank you.